Hello and welcome everybody to my channel ominibioentrance, and today we will be seeing CSIR PYQs June 2023 Shift 2. I will be dealing with applied biology and methodology, DNA replication, protein synthesis and genetics. These are solved questions of part C. If you are new to my channel, please do like, share and subscribe. I have also uploaded old CSIR PYQs. You can refer to my channel for the same. Then next one. The following statements are made about post-transcriptional processing. RNA editing can occur via the deamination of cytosine residues leading to formation of uracil and thus a change in the coding sequence. A is correct. The major spliceosomal complex mediates removal of group 2 introns. Group 2 introns are self-splicing, so they don't require a spliceosome, so B is not correct statement. Trans-splicing events seen in trympanosomes allow the formation of multiple gene products by bringing together different combination of exons of 3 or more genes. It is not 3 or more genes, it is 2 genes, 2 different primary RNA transcripts. So C is also not correct. Capping of eukaryotic mRNAs occurs exclusively in the nucleus of the cell. It can occur even in the cytoplasm of mammalian cells and trympanosomes. So D is also not correct. So the answer here is A only. Option 4 is the answer. Then column X and Y of the following table lists some treatments, methods, reagents and events that are related to human lymphocyte culture and banding or karyotyping of human chromosomes. Which one of the following options represent all correct matches between column X and column Y? 5% barium hydroxide treatment at 50 degrees Celsius. So that is for C banding, A2. Trypsin treatment is for G banding. Phytohemagglutinin is for mitotic stimulation. Phosphate buffer treatment at 80 degrees is for R banding. And silver staining is for nucleolar organizer regions, NOR. So the answer here is option 1, A2, B4, B5, C3, D1, E4. Then next one. Given below are some statements that are associated with transgenic plants. Each statement has a blank space indicated by a dash. You have to fill up the blanks. Which of the following options has the correct sequence of terms that can be used to fill in the blanks in the above statements from A to D, such that all the statements become true? A transgenic plant has 2 functional copies of a transgene can segregate in a 3:1 ratio. So 1 and 2 options cannot be considered. We have to see for 2 and 3. The dash system can be used for the removal of marker genes from transgenic plants. Krelox can be used. B. The endogenous plant gene dash can be used to engineer resistant to imidazolone herbicides, that is ALS. That is acetolactate synthase is inhibited by imidazolone herbicides. So option 3 becomes more correct. Then variations in transgene expression levels between 5 independent transgenic lines generated using the same tDNA construct can be due to position effect. So D is the right, D is position effect. So the answer is options 3, that is A 3:1, B Krelox, C ALS and D position effect. The figure below represents the data from immunoblots, IB. So whatever IB, beta-catenin, IB, E-cadherin, that is all immunoblots. IB represent immunoblot. Here they have given in the question IB is immunoblot of co-immunoprecipitation, IP beta-catenin, IP beta-catenin, Okay, so IP is co-immunoprecipitation experiments with antibodies as indicated after HeLa cells were treated with arsenic. B-cat refers to beta-catenin, E-cat refers to E-cadherin. Now, before coming to answering the question, I will just give a basic of WNT signaling. So WNT signaling pathway, it is of two types, that is canonical and non-canonical form. Canonical lead to regulation of gene transcription and non-canonical is of two types, that is planar cell polarity pathway and WNT calcium pathway. WNT calcium pathway regulates calcium inside the cell. Calcium is required by the E-cadherin. Now planar cell polarity pathway regulates cytoskeleton. So non-canonical pathway we have got E-cadherin. Now when there is the canonical WNT pathway causes accumulation of beta-catenin in the cytoplasm, that is over here, this diagram. It causes accumulation of beta-catenin in the cytoplasm and its eventual translocation to, this is the nucleus, eventual translocation to the nucleus to act as transcriptional co-activator of the transcription factor, that is TCF, and resulting in the transcription of DNA, which finally result in cellular proliferation. So E-cadherin is for non-canonical pathway. And finally, when beta-catenin enters into the nucleus and is bound to TCF, it results in cellular proliferation. Remember that also. Now coming to the question. Now in arsenic negative condition, only very little beta-catenin is present in the cytoplasm. You can see over here, this is cytoplasm, this is the blot. Only very little beta-catenin is present. In B also, only very little beta-catenin is present in the cytoplasm. Now whatever is present in the cytoplasm, this must be what was already present in the cytoplasm even before the experiment. No, sorry. What was already present in the cytoplasm of HeLa cells. So that must be that beta-catenin which is present in the cytoplasm. Now input is your control. They have given input, that is your control. So let it be there. In control, beta-catenin is present. Then E-cadherin is not present in the arsenic minus condition. This is E-cad. Immunoblot of E-cadherin in arsenic minus condition, E-cadherin is not present. Now, when arsenic is added, beta-catenin starts accumulating in the cytoplasm. When arsenic is added, beta-catenin starts accumulating. Here the band is darker for beta-catenin and it starts accumulating in the cytoplasm. E-cadherin also starts accumulating in the cytoplasm. Here also you can see E-cadherin getting accumulated in the cytoplasm. In the absence of arsenic minus, we saw that beta-catenin only a slight band in the cytoplasm. TCF4 is not showing the beta-catenin band here. But in arsenic plus condition, TCF4 is showing the beta-catenin band. Why? Because it shows that beta-catenin got localized to the nucleus and it got bound to TCF4. That is why beta-catenin band is seen in TCF4 immunoblot showing co-immunoprecipitation. And this results in cellular proliferation. That is what diagram I have showed earlier. So arsenic stimulates WNT signaling pathway and also E-cadherin is also stimulated. So it acts on the non-canonical pathway. Now we will read the statements. Arsenic activates the non-canonical beta-catenin pathway. That's correct. That is why it stimulates the WNT signaling pathway and also E-cadherin is also stimulated. So it acts on the non-canonical pathway. So that is correct. Then arsenic lead to cellular proliferation. Yes, that is why we got beta-catenin got bound to TCF4 in the nucleus and it results in cellular proliferation. Arsenic lead to proteasomal degradation of beta-catenin. No. Beta-catenin is increasing, accumulating in the cytoplasm on addition of arsenic. So it is not leading to proteasomal degradation of beta-catenin. Arsenic induces apoptosis in the HeLa cell. No, it is causing cellular proliferation. So A and B are the correct answers. So answer is 1, that is A and B.